Hello, and welcome back to Human Evolution. Today I want to talk about DNA, or the Instruction Manual of Life. The first thing I want to do, though, before we do that, is I want to remind you about the four major biological macromolecules. While we're going to be talking about nucleic acids today, and we'll be talking about proteins in the very near future, we will not spend a lot of time talking about lipids or carbohydrates, although in reality, we do talk about them when we talk about respiration, when we talk about cell membranes. Because remember, the plasma membrane is actually made of a phospholipid bilayer, right? Well, lipid is that kind of molecule. Glucose that we make in photosynthesis and that we use in respiration. We don't photosynthesize, by the way. I just said like we in general in life, but that is used in photosynthesis, and then broken down in respiration. That's a sugar, right? So carbohydrates are your starches and sugars. Lipids are your fats and waxes. Proteins do lots of things that we're going to talk about. And nucleic acids are your DNA and RNA. And that's what I want to talk about today. Mostly DNA, a little bit RNA, but we're going to talk about RNA specifically in the future. Okay, so nucleic acids are made, the building blocks, right, are made of nucleotides. Those are the building blocks of nucleic acids. And a nucleotide is a molecule that consists of a phosphate group, a 5-carbon sugar, which is why it's called a pentose sugar. So you've got your 5-carbon sugar, and then you have your nitrogenous base. And it's the nitrogenous base that's like the functional group. It's the interesting part. So if you think back to chemistry when you talked about amino acids or things like that, which we will talk about later on, you always had that functional group. And so this is kind of what makes the nucleotide the type of nucleotide it is, right? So this is the key to both DNA and RNA. So you can see this is a DNA nucleotide and this is an RNA nucleotide, so a deoxyribonucleotide or a ribonucleotide, because remember DNA is deoxyribonucleic acid and RNA is ribonucleic acid. So here you've got your phosphate, you've got your sugar, and you've got your nitrogenous base. Phosphate, sugar, nitrogenous base. The big difference, though, is that you have an extra oxygen on that sugar. So the difference between DNA and RNA in terms of the structure of the nucleotides, that's going to be in that sugar. And you can see right there, okay? So, nucleotides bond together to form the structure that we know so well and the structure that we love of DNA. What kind of bonds do the nucleotides bond with, right? What are those bonds that cause those base pairs to form? Are they covalent bonds, ionic bonds, hydrogen bonds, or magnetic bonds? So think back to chemistry, organic molecules. They're going to be hydrogen bonds. So when you look at this wonderful structure, what you're really seeing is two nucleotides across from each other. So here's one nucleotide, here's another nucleotide, and here you actually can see they've made little boxes for the nucleotides. Here's one, and here's an actual picture of DNA, and you can see that beautiful double helix in it. This is bacterial DNA. You can know that by that wonderful characteristic ring that I was telling you about. Okay, now this characteristic shape of the molecule of DNA is called a strand, a staircase, a double helix, or double spiral. Well, it's called a double helix. And so how does the double helix form? Well, it has to do with the nucleotides. The nucleotides, there are 4 of them. They are adenine, guanine, cytosine, and thymine. And they come in 2 different categories, and you can see this based on their shapes. You have this double ring here, adenine and guanine share that. They are purines. And then you have cytosine and thymine, which are these 6-carbon sugars, and they are called pyrimidines. So you can divide your 4 nucleotides into these 2 categories, purines and pyrimidines. And this is it. All DNA is based mostly on these 4, well, all DNA is based on these 4 nucleotides, A, G, C, and T. So, when you look at the genetic code for an oak tree, would it be more similar to an ash tree than to a squirrel, more similar to a chipmunk than a maple tree, more similar to a mosquito than to an elm tree, or would it be exactly the same as an octopus? Well, remember, the genetic code is those 4 letters, and it is universal for all life. The genetic code is the same. If you just pull out a strand of DNA, you wouldn't be able to tell if it was an ash tree or a squirrel or an octopus or an oak tree or a mosquito or a chipmunk, right? You wouldn't know because it's made up of all those 4 letters. They are the letters of the words of life. So when we look at DNA, what we're really talking about is this collection of nucleotides, right? Deoxyribonucleic acid is this instruction manual, right? It's the words. So when I talk about specialized cells reading certain chapters, I'm talking about they're only looking at certain parts of the DNA because we know that every cell has all of the DNA. It is double-stranded as opposed to RNA, which is single-stranded. And in DNA, because it is double-stranded, each strand has a hydrogen bond bonding it together. But here's the thing. The ladder part, so the rungs of the ladder are the nucleotides, but the sides of the ladder of DNA are going to be this phosphate backbone. And what that means is that the phosphate on the nucleotide will bind to the sugar on the next one. So you always get this chain of phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. And this is true whether it's DNA or RNA. Now you might notice over here that I've got a bunch of nucleotides listed and some of them say RNA. We'll be talking about those when we actually talk about RNA. Okay, so we know that DNA forms these strands, as does RNA. But DNA binds 2 strands together. How does that work? Well, we know it's going to be through hydrogen bonding, but they're going to bond in a very characteristic way. If you'll notice, this ladder is very smooth, right? It's the same width all the way down. But didn't we say that there were actually 2 kinds of nucleotides and one was bigger than the other? The purines are bigger than the pyrimidines. So if you were to have 2 purines bond together, you would have a little narrow spot in your DNA. And if you had 2 pyrimidines bind together, you would have a wide spot in your DNA. By the way, I'm going to go back a slide just for a second to this one. An easy way to remember which your purines are and which your pyrimidines are is the phrase cut the pie. So CT, cytosine, thymine, and there's a U in there, and we'll talk about the U later. But C and T, the PY, cut the pie. So cytosine and thymine are pyrimidines. That's an easy way to remember it, and it will help you know when you're looking if you're binding correctly, right? So the bottom line is this. Your pyrimidine, right, thymine, can only bond with adenine, which is a purine. The purine guanine can only bond normally with cytosine, which is a pyrimidine. So all of your bonding will be T's and A's, C's and G's. And the easy way to remember this is that the straight line letters bond and the curvy letters bond. Okay, but what that also means is that you should have the same number of C's and G's, right, and the same number of T's and A's. So if I have a strand that has 30 nucleotides in it and 10 of them are thymine, that means 10 of them have to be adenine. Well, 10 plus 10 is 20. My strand was 30. That leaves me only 10, so only 5 can be guanine and 5 can be cytosine, right? Because you have to have the same numbers of each of these because they make a pair. So to illustrate that point, let's look at which of these samples supports these base pairing rules, okay? So in sample A, 35% of your sample is guanine, 35% of your sample is cytosine, 15% of your sample is adenine, 15% is thymine. First of all, do these actually add up to 100? 70, 30, they do. So that's good. In sample B, 40% is guanine, 10% is cytosine, 40% is adenine, 10% is thymine. Do they add up to 100%? They do. And then in C, you have 25 of each. I would like to point out right now that C supports our base pairing rules and also does not. So C does support our base pairing rules, but it doesn't tell us very much at all because it supports other base pairing rules too. So really A and B, so C is correct, it's just not good. But out of A and B, one of them is going to say, yes, that supports the fact that C's and G's bind together and A's and T's bind together. So which one is it? Well, let's think about it. If I have 35% guanine, and I know that guanine bonds with cytosine, then I have to have 35% of cytosine. I do. And then that is 70%, so I only have 30% left over for my ATs, right? Well, then I just split it in half. 15% is adenine, 15% is thymine. Okay, that does support our base pairing rule. 40% guanine. Oh, wait. G and A, they're both 40%. Can those have the same number? Can two purines bind together? Can two pyrimidines, C and T, can they bind together? No. B does not support the rule. Okay, B does not support the rule. So A absolutely does. C does, but doesn't tell us anything. Okay, so that's a little bit about DNA. We've got our alphabet of 4 letters. They bind together tightly between the phosphate and sugar, phosphate-sugar backbone. So it's called the phosphate backbone, but it's bound to the sugar. Then we've got our nucleotides that stick out and make the rungs of the ladder of our double helix. Now, the other thing that we want to talk about is how do we copy it? Because any time we have a cell that's going to divide, don't we have to have new DNA for that? Of course we do. So DNA is commonly referred to as the hereditary, excretory, regulatory, or proteinaceous material. Well, it's hereditary because each cell gets a new copy. Each cell gets a new copy with all the instructions. And when we talk about differentiation, we talk about just reading some of those instructions and ignoring the rest, but they're all still there. So, hereditary. Okay, so we already know why DNA replicates, but don't forget, it always replicates in interphase, in the S phase, right, of interphase, in that synthesis phase. And then we'll either undergo mitosis or meiosis, depending on the cell. We know that most cells undergo mitosis, yay, right? But we've got this DNA synthesis that's going on here. And remember, every cell has to have a full complement of DNA. So how does this happen? How do we replicate? Well, first of all, it's in that double strand. The very first thing that we have, or it's that spiral, right? That double helix. The first thing that we have to do is unwind it and unzip it. So enzymes, which we're not going to name the specific enzymes, but this is where you have enzymes like helicases and topoisomerases. We're just going to say enzymes and cluster them into a little category together. Those enzymes unwind, they unzip, right? And one of the big things that they have to do is make sure that you don't get weird coils later on. But that's what's happening here. Then you're going to have some proteins that will stabilize that unwound DNA so that it can be replicated. You got to hold it open, right, essentially. Okay, now once it's held open, you have these half strands that can have new nucleotides added to them, right, so that you get new strands of DNA. So for every one original strand, you can get 2 new ones. And the interesting thing about this is what's called semi-conservative replication. And what that means is that each new strand is half old and half new. So it's semi-conservative. The original strand isn't conserved, and you don't get a brand new one. You get 2 hybrids, old DNA and new DNA, and that's called semi-conservative. Okay, so now 2 different things have to happen here because it's important to note that DNA can only be read in one direction. It's read from the 3' to the 5' end. So if you're wondering where the 3' and the 5' ends come from, that comes from the orientation of the sugar. Remember, I told you that each carbon had a number assigned to it. So what that means is that you have a phosphate group and a 3' on this side, and then it's oriented so that the 5' of the sugar's on this side. Then you have your phosphate group, 3', 5', phosphate group, 3', 5', 5'. Right. Okay, so If you can only read it from 3' to 5', that means that it has to grow on the 3' end, because when you have a strand of DNA, those two nucleotides, they will be flipped from each other. So if this side, because they're opposite, right? They're on the opposite side. So if you have 3' phosphate or 3' 5' phosphate going in this direction, on this side, you'll have 3' 5' phosphate going in this direction because it's opposite. Now, your growing end of your replicating strand is always the 3' end. So if it's reading from 3' to 5', if you're on this top strand right here, which is opened, right, 3' to 5', it's unzipped that way, then the DNA polymerase, and the DNA polymerase is a protein that is a copy machine, essentially. And well, it's not exactly a copy machine. It's the thing that it's a synthesis machine. It's like a little factory. That's better. It's like a little factory. And it allows nucleotides to pop into it, and then it sticks them right next to each other and across from the correct nucleotide. So it places it and it just adds them in, right? It can just zip along 3' to 5' towards this replication fork. And remember, the replication fork is the unzip part. And this actually looks just like a zipper. I mean, imagine it works just like a zipper. So you can just go zoop right down it. This is not true for the other strand, which is called the lagging strand. So the leading strand, you just synthesize continuously. Yay! Our little protein called DNA polymerase, our little new strand maker, is having a great time. The new strand maker, the DNA polymerase, on this side, having a terrible time, because as it opens, it's opening from 3' to 5'. And so if the polymerase were to start here at this like second little stabilizer, if it hooked on here, it could replicate the rest of it, but as this unzips, it's not replicating any of this part. So the lagging strand is synthesized what we call discontinuously in these little fragments, right? And those fragments are called Okazaki fragments. They're named for the man who found them. So you'll have multiple polymerases that will hook on this lagging strand, and they will just replicate until they hit the previously replicated strand. Now the problem with that is that there's a lack of connection there, because remember, they were synthesized at different times. So you have to have another protein called DNA ligase that will hook on. It'll seal this cut, right? It'll seal the two fragments together, and when the ligase is done, you will have a nice full strand. And so it works. It's just a little bit trickier than the leading strand. So leading strand is easy, boom, 5' to 3', continuous. The lagging strand has to have a little bit of help. It's done in pieces, okay? Names of things you need to know: DNA polymerase, that's what actually does the making of the new strand. DNA ligase hooks them together. Okazaki fragments are the discontinuous fragments on the lagging strand. You need to know the name of the leading strand. You need to know that we're read 3' to 5'. We grow on the 3' end of the new strand, and that a bunch of enzymes proteins are responsible for zipping, or for unzipping rather, and stabilizing the opening strand. And that is DNA replication in a nutshell. And here is your last question. So understanding all of this, right, why are we able to take a gene from one organism, put it in another one, and have it function just the same as it did before? Is it because all organisms are made of cells, all organisms have nuclei, all organisms have ribosomes, or because the genetic code is universal? Well, remember, I told you if you pull out a jellyfish gene, you won't know it's a jellyfish gene, right? It's just DNA. The genetic code is universal. So the things that we've learned about today and the things that we're going to learn about in the future are going to build up to a conversation about biotechnology and how we manipulate genes in the future, like how we do and will manipulate genes, right? We can do this because the genetic code is the same for every living thing. It's actually very cool. All right, so if you have any questions, please let me know at the beginning of class, and thanks for listening. I'll see you next time.